if anyone's out there. Nope. All right. It's not bad. We got 10. That works. All right. Yeah, we got people watching. That's always good. All right, so exams next Friday. Believe me, I don't want to make the exam. This will hurt me more than it hurts you, right? I've been trying to make the questions as I've been doing these quizzes, so it shouldn't take me as long. That exam one took me darn near 3 to 4 days to get it done. And then Blackboard is not friendly, that's for sure. All right, so we've got the exam on Friday. That is covering this week's lab, so the marker capture stuff will be on the exam and on exam 1. Next week's lab we'll collect data. That is not going to be on that exam. All right. I'll have to go back to see what we cover. I believe it's the diversity stuff. So that was 2 labs of diversity, and then this marker capture. I think it's just those 2 labs. So kind of plan for, I don't know, 2 questions for each. 2 to 3 questions for each. Kind of check your concepts. All right, so this is where we left off. Freshwater systems review. Just kind of rehash what we're going to do in preparation for moving towards the marine environment. All right, so when we deal with the freshwater system, our fundamental separation is whether or not that water is flowing or standing still, because that has a big difference in what organisms can live there, namely the type of photosynthetic organisms. It's much easier to have floating algae, this phytoplankton, in a system that is not moving, in a lotic system. So this phytoplankton in these lake systems is the primary source of production. In a flowing water system, streams and rivers, those floating algae are just going to get swept away downstream. So the primary source of production in those systems will be any of the algae that's attached to structures. And then we call that periphyton. All right. And then these systems differ in how they interact with the terrestrial system. In a lake or pond type system, you look at the watershed and you can have nutrients sweep into those systems from the terrestrial system, but mostly those ponds and lakes get most of their energy from this autochthonous sources. So the primary production, taking the light energy, making it into sugars. Streams and rivers, on the other hand, get most of their organic material from the terrestrial system itself. So those are allochthonous sources. All right. Just so happens that as you get down the river system, as you move down that continuum of the river and increase your stream order, you're starting to collect more and more of that terrestrial runoff, the organic particles and so forth in those rivers and streams. All right, and then we talked about like stratification and so forth. Any questions on freshwater system? Because our marine system also exhibits zonation and has various names, but the ocean system is quite different. All right, so when we look at the marine systems, we're going to base our communities, base the physical factors and stuff on the depth that we're interested, depth that we're studying, the bottom profile that we're studying, as well as the species that are there, that are present in that area, or which basically the species that can survive that area. We can separate the different environment based on zones, much similar to what we can in the freshwater system. All right. But in a marine system, we can actually add a few more zones. So in a freshwater, we talked about the littoral zone, the pelagic zone, the profundal zone, benthic zone, photic zone, and so forth. So all of those are there, but we can delineate a little bit more in the ocean to include more things like the neritic zone, the mesopelagic zone, and so forth. All right. I have this figure here with all the zones, with all the marine zones on here as more of like a FYI. I'm not going to ask you for the marine zones. We're not going to get into individual details, but I do think you should know the zones for the freshwater system: littoral, pelagic, benthic, profundal, and photic zones. All right. So in our marine system, what distinguishes it from freshwater, hopefully, is it's obvious, and it's the salinity. All right. So why is the ocean system saline? Why do we have salts in there? Well, it's derived from a couple different sources. Some of that source comes from terrestrial system. So rain falls, your water cycle, your hydrologic cycle has precipitation falling on the land. The water interacts with the soils, and as it runs off and it runs into aquatic systems, perhaps in aquifers, it dissolves and takes up minerals, and then those minerals and salts go into the ocean, ultimately get to the ocean. All right, so you do get some salts from terrestrial runoff. Majority of that salt, though, is due to interactions of the water with the underlying parent material. So the rocks and the soils in the ocean itself, what underlies that ocean, that's where the majority of the salt in the marine system comes from. All right, how much salt are we dealing with? It ranges. It ranges between 3.1 and 3.8% salt. All right. In other books, you'll see it as an average of 31 to 38 parts per thousand, which is this 3.1 to 3.8%. Our average is about 3.1. I'm sorry. The average is about 3.5%, 35 parts per thousand. That's our average. But we do have variation. So why do we get this variation? Well, we get variation when we have rivers, freshwater input. So the rivers are bringing fresh water into the system. Saline, the salt concentration is going to fall down in some of those areas. All right. We have areas that are hot and dry and exhibit a lot of evaporation. In those areas, you're going to see elevated levels of salinity, higher levels of salinity. All right, upwards of 3.8%. You have some areas, let's say the Arctic areas, where ice forms. All right. In those areas, you often get higher levels of salinity right near the surface, right where the ice is formed. All right, so we do have ranges. The average is about 3.5%, but by and large, this is definitely saltier than a freshwater system. And organisms have to deal with that. Now, in our oceans, we have something called a biological pump. This biological pump takes carbon from the atmosphere, gets it into the water, gets it integrated into organisms, and then gets transferred throughout the ocean. All right? And we have this pump. This pump is important because it's a way to get carbon down to the depths. All right? You can't rely on diffusion alone to get CO2 down to the depths of the ocean. And it's just not going to be possible. So in these, and I'll say, the same thing applies to the deep freshwater lakes as well. You're going to have a biological pump there to get the carbon transport down to that area. So what is this? You have your dissolved oxygen in the water, your phytoplankton, other algae is going to take it up, it's going to fix it, it's going to assimilate it, integrate it into sugar molecules and ultimately into organic tissue. All right, and then that tissue, or I should say, those compounds get eaten by herbivores, which then integrate it into the tissue. Those herbivores get eaten by prey or by predator, and so forth and so forth. So eventually what happens is that you have this buildup or movement of carbon down into the predators. You also have dead organisms that will sink down to the bottom, providing and taking carbon down to the ocean. All right, so this distribution movement of CO2 in the oceans is driven by the biological pump, and it's aided by our ocean currents. So it's not going to be just dropped down into one area. Our currents are going to move that throughout the ocean itself. And our oceans represent a huge carbon sink. You've probably heard it, you've probably seen it. It talks about, well, the oceans are going to take up any excess CO2. It takes up some of it. I mean, it will take up some of it. And it is one of the major carbon sinks. If you have outgassing of CO2 from the oceans, that would be a huge amount of CO2 that would be lost. Is that likely? No, it's not. It's not likely at all. So what about our environment? What about the variation in our ocean? So we already talked about variation in salinity. What about temperature? What about everything else? Well, temperature is going to vary based on solar radiation in general. All right? So the closer you are to the equator, the warmer the waters are. As we move towards the pole, the waters get colder. All right? But that gradient isn't going to be a very nice gradient like we have in a freshwater system. All right? We see variation because of ocean currents. All right? So the ocean currents help to modify or modulate some of these relationships, some of these trends in temperature, and some of the other properties such as oxygen or salinity and so forth. All right? So what are these currents? What generates them? Well, the currents get generated by 3 different processes. The major driver is the Coriolis effect. What is the Coriolis effect? It's the spinning of the Earth. All right, so as the Earth spins, you're having the ocean that kind of stays stationary, and that generates the current. All right, I'm going to see if I can find something here. See if we can find it. All right. Let's see how long these are. I don't want to watch a whole lot. All these different kinds.
Hey, it's me, Des. Welcome back to Smarter Every Day.
Check this.
We're in the rocket van, and I've got 2 little science helpers here, right?
All right. We're going to mute this because we're not going to watch the whole thing. So what this is dealing with is he has a weight to just kind of demonstrate what's going on. Open that bigger. All right, so you have the weight in the car, and as you drive, right, the weight's going to drift back. You feel that. I mean, you see it with the with coffee when you drive, the coffee kind of sways back and so forth. You stop. But what happens with a helium balloon? And this balloon kind of demonstrates this whole Coriolis effect as to what happens. So you can imagine the van, the stationary part of the van as the Earth, and the Earth is going to be moving on it as it rotates, and the water is this helium balloon. You see what happens? It moved forward. Why did it move forward? So it initially moved forward when he started. I'm going to rewind that. You see that? He started going forward, and the balloon moved forward. That's opposite of any liquid. So why is that? The van moved forward, and you have this air mass in the car, and the air mass drifted back and then came up forward because the air has to go someplace. So mass hits the back, gets pushed forward, that causes the balloon to move forward. That is the same thing as what's driving these currents, and it explains the direction. So our Earth is round, it's a little bit bulged, all right, so it's going to be a little bit, it's not a perfect sphere. So as the Earth rotates, and we're rotating in this direction, so this direction, it's causing the water to kind of stay put, and it forms the currents going in the backward direction, right at the poles, or right at the equator. Once these currents hit the land masses, they have to go someplace. So they're going to go around. So they're going to go north or they're going to go south. All right? And along that time, as we're moving, these surface waters are picking up warmth. All right? They're absorbing heat from the sun, which means then these currents are going to be warm water currents. All right? They're moving. But something has to be replaced. As we're moving this water in one direction, the water has to be replaced. And where does it get it? It's going to get it from other locations. It's going to get it from the depths. It's going to get it from more of the northerly water masses. And those water masses are cold. So even though we're at the equator here, or we're at the equator and we're in the tropics, these are cold water currents. You go to California, these are cold water currents out on the coast, even though our land temperatures are very warm. All right? So the Coriolis effect is only one part of it. We also have temperature effects and we have salinity effects. And that's going to drive a lot of these currents too. So salinity effects. What's our density of salt water compared to fresh water? Is salt water more dense or less dense? Salt water? Yeah. Yeah, you can put egg in salt water and it floats, right? You put an egg in fresh water, kind of drops, right? Yeah, so it's easier. So salt water is going to be more dense, all right? So if you have a spot that has higher salinity, all right, it's going to cause it to try to float down a little bit more, and you can generate more of a current, more of a vertical current with the differences in salinity, all right? Same thing applies to temperature. Colder water is more dense, all right? So the colder water can tend to drop more than the warmer water. So the Coriolis effect can kind of produce your lateral movements in the water. Your temperature salinity differences can start to set up more of your vertical movements, your vertical currents. And wind also plays a role, but not so much in these overall major currents. All right, so as these currents are happening, right, water has to be replaced, water has to go someplace. All right, so that can lead to upwellings and downwellings in the oceans. All right, so the upwellings are places where you have nutrient-rich water being brought to the surface. This water is cold water. So in our systems, you have, we're going to use the South America. So we're at landmass of South America. Say that's our Andes Mountains, all right? We have our current flow moving that way, all right, because our globe is moving that way, all right? This water mass has to get replaced. All right, so where does it get replaced? It draws the water up from the depths to take its place. This water is cold. And nutrient rich. It's cold and nutrient rich. That is a site of an upwelling. What kind of diversity do we have at an upwelling? We actually have a lot of diversity. These are sites of high diversity. Okay, sites of high diversity. This is an example of an upwelling. You can see, and what we've done is they've colored photosynthetic production. And you can see how much higher we have right in these spots. This is a Galapagos. All right? You've got these nutrient-rich waters driving high amounts of primary production, which then can support more herbivores, which then can support more predators. You just have this big boom in biodiversity at those sites. All right, so we've drawn this cold nutrient water. This cold nutrient water, I mean, the nutrients are down there. Why are the nutrients down there? The biological carbon pump. It's taken the carbon down to the depths. And these upwellings are a way in which we can bring those nutrients back up to the surface. Downwellings are the opposite. Now, with our downwellings, we're going to lose our nutrients. We're going to lose some oxygen. The reason we're losing the nutrients is because you have things that are dying and being taken down to the depths. On their way down, you have decomposition, aerobic decomposition, and that's going to suck up some of the oxygen in those areas. Now, some of that is at our opposite end. So here we can say we have Asia. All right, our globe is still rotating. We have our surface here. All right, that water has to go someplace. Now, normally it's going to kind of hit and kind of go north and south, but in this area, we have huge river input. So this is our river. So we have a huge freshwater input going in there. All right, that's bringing in nutrients. All right, it's bringing in nutrients, and that difference in salinity then is going to start causing water to move downward in a downwelling. All right, it's going to start moving downward in a downwelling. And also, as the water cools, you increase your density. That also helps promote these downwellings. So our upwellings are sites of a lot of nutrients, a lot of biodiversity, a lot of primary production. Downwellings, though, we often see diversity. We often see high diversity, but with that diversity, we're taking nutrients away from our ocean system. We're locking them away down into the depths, and we're depleting oxygen. All right. So most, but not all of our abiotic factors are going to change with depth. This is the same thing in a freshwater system. If you increase your depth, you're changing the light intensity and the wavelengths of light that penetrate it. Temperature, you increase in depth, you're going to get a decrease in temperature. All right. Salinity, you can have salinity differences due to differences in salt and let's say more saltier water and less saltier water. All right, pressure has an impact. I mean, pressure, if you've dove into a deep pool, I mean, you felt the pressure. Organisms have to deal with that, as well as oxygen content. So, we can't really set up generalities because it varies based on where we're talking, where we're at. Are we at an upwelling? Or are we at a downwelling? Or are we in tropical shallow areas? Are we in polar deep areas and so forth? So some of these things are going to increase, some of them will decrease, and it's just going to be specific on where we're at. All right. A lot of those variations are going to be driven by the currents themselves. So the currents can help bring increasing temperatures or colder temperatures or nutrients, up to the surface and so forth. But regardless of all of these factors, the main driver of all of our production, of all of our diversity, is going to be that primary production. All right? So it's going to be that area of the photic zone where our algae can grow. Both our phytoplankton and any of our attached algae and other aquatic vegetation. Because of this, because all of our production is driven by the photic zone, or is being driven in this photic zone, the most variable communities that we see are those that are near the shore, those close to shore areas. They're in the photic zone, you're going to have the primary production, and in those areas you're going to have a tidal regime. So some of the areas will have, will become dry, or almost dry for a part of the day, and then it'll be flooded and so forth. You have variation in salinity gradients because of freshwater inputs and so forth. All this variation that happens in our physical conditions will also translate to variation in our communities. You go out to a deep ocean, you go out to the abyssal zone, the community, yeah, there's going to be some diversity there, but it's not really going to be highly variable. Those deep parts in one part of the ocean versus deep part in another part of the ocean, they're going to be basically similar. But if you go to a mud flat, on the East Coast and compare it to a mud flat on the West Coast, you're going to see differences in our community. All right, questions on the marine environment. This image is just another image of our biological pump. Your book talks about some of those surface currents. So those are also a way in which we can kind of mix in our top layer. and also gives you a little bit more idea as to why you have wind blowing water in one direction, blowing surface water in one direction, but we have deeper water moving in a different direction. So if you're interested in that, check it out, or you can take oceanography. I think oceanography is being offered this spring. All right, aquatic environments. Light availability varies by depth. All right, number one thing in water. That's the number one most important thing in water. All right, that availability is the light intensity and our wavelengths. All right, that's what changes. It's this the light availability that is, at least in part, determines our zonation. All right, photic zone versus aphotic zone. What causes this decrease in availability and wavelengths? It's the absorption and the scattering of light by the water molecules and by anything else that's in that water. All right, the carbon in our system comes from the atmosphere. CO2 is going to dissolve into our water. All right, it's going to be picked up by our algae and then can be transported down to the depths with the through the biological pump, or it can be transported someplace else just by the currents. Streams and rivers are our lotic system. They are defined based on the water flow, all right? And their communities vary based on stream order. All right, so as we go from a small headwater stream to a large meandering river, all right, our physical and biological characteristics will change. And it's tied to those organic particles, of course, particulates versus fine particulates, as well as what type of primary production and amount of primary production that occurs along that continuum. Ponds and lakes basically have the phytoplankton as their primary drivers, all right? But in these lakes, stratification becomes important. So over the summer months, as it warms, you can get a lake based on two different layers, epilimnion above, hypolimnion below, and that border, that transition, is the thermocline. That stratification can limit our water movement, all right? Can prevent oxygen from getting down to the depths. It can lock nutrients away, which is why we rely on turnover events to get it mixed again. Replenish our oxygen and distribute the nutrients. In the marine environment, salinity is the primary difference between this environment and our freshwater. All right. Now we say it's 3.5%, but it actually varies. All right? It varies from about 3.1 up to about 3.8, and the reason it varies is because of freshwater input, differences in evaporation, as well as some ice. So the polar environment's a little bit different. All right. Now in our oceans, we do have complex patterns, both local and regional diversity. All right, and it's tied to the currents. All right, so the currents could be warm, they could be cold. All right, could be nutrient rich, could be nutrient poor. All right, they're driven. These currents can be horizontal, they could be vertical movements. All right, and these currents are really caused by movement of the Earth. That's the Coriolis effect, temperature differences, and salinity differences. All right, and all of these things, when you factor in all this variation, you have to remember our organisms that live there have to be able to deal with these conditions. All right, they have to be adapted to it. The law of tolerance states that they can only survive in the areas where they can tolerate. Questions? All right, so think of questions for this as I prep our next presentation. All right, if we don't have questions, we will move on. All right, so we talked about environmental variation, the drivers for the variation that we see. So discontinuities in our physical environment. All right, so our environmental is caused by these discontinuities, and our organisms have to deal with it. So these discontinuities is what drives the variation that we see across the species. All right, so what we can recognize is that our habitat, our area that we're interested in, actually consists of patches. And it's these patches where our organisms can live. Question is, how do the populations respond to this variation? Should they respond genetically, evolve adaptations, or should they take more of an approach of, let's say, phenotypic plasticity and say, well, let's wait to see where we are and then respond accordingly? All right, this is the intraspecific variation. So we're not going to deal with communities, we're going to deal with populations. We're going to deal with the species and look at what causes the variation across the species. All right, so intraspecific variation does exist. We know that. All right, and what's our definition of this? And I provide it just so you have it. But intraspecific variation is any genetic or phenotypic variation within a species. So our species is going to be those individuals that can mate. All right, I do have a definition of a species later on. I can't remember if it's in this presentation or if it's in the next one that I'm working on. All right, but this variation is going to be any genetic or phenotypic variation. So you can have genetic variation without seeing a difference in phenotype. All right, that's still variation that we're interested in. Now, what causes this variation? It's going to be influenced by both the spatial and the temporal variation that our species encounters. All right, could be encountering over evolutionary time span, or it could be the variation that it sees today or yesterday. All right, so the spatial variation that we're interested in looking at is going to be the magnitude of the differences between our habitat patches. So the different habitat patches where our species can live can have different populations in them. How big a difference they are can lead to this intraspecific variation. How isolated those patches are can lead to intraspecific variation, as well as the relative sizes of these patches, big patches versus small patches. But spatial variation is only one aspect leading to this intraspecific variation. We also have to account for the temporal variation. So how does this variation, this environmental variation, change through time? Is it a regular variation? Is it a fast change or a slow change? Is it predictable or is it unpredictable? So these are the things that we're going to talk about. All right, so what we're going to lead to is kind of talk about, we're going to start with the spatial variation, then move to the temporal variation. I'll let you get done writing this. This is definitely a text-dense slide, right? Get it? It is. It's a lot. All right, so we're going to start with the magnitude of differences between the patches. So I'm going to say, as I kind of make this, I've got notes and how I outline my notes and so forth. So kind of a new idea. So spatial variability. All right. First thing, magnitude of differences between our patches. So our species exists as populations in these different patches. Maybe they're a subpopulation, maybe they're an entire population. All right? Our fitnesses are derived based on the environment in which they live. So think back to the 3 properties of fitness. What are the 3 properties of fitness? What was one? property of fitness. What's that? Genetic. Okay, so fitness is determined by the genotype, right? So if you have the same genotype, you should have the same fitness. Property number 2. That's what this slide is about. Yeah, it's specific to the environment. So we have, if the environment changes, our fitness is going to change no matter what. Our genotype didn't, but our fitness has changed because the environment changed. And then our third thing, less tangential to this, you have to measure it over more than one generation, right? So we can't just go out into a population and measure it. We actually have to look at the reproduction and so forth, measure it in offspring and grand offspring. All right, so environment determines our fitnesses. The environment also determines then the selection pressures. So the more similar our patches are to each other, the more similar our selection pressures are. And that means you're going to produce less intraspecific variation in our species. If you have 2 patches that are drastically different, but our species can survive in both, well, each patch is going to have its own set of selection pressures, and that means we have our own evolutionary trajectory in each path, in each patch. How do you think that's going to affect our intraspecific variation? Should increase it, right? Should increase it. So the larger the difference is between our patches, then the more differences that we should see. However, we have to account for that boundary layer. So the boundary layer between our patches is called the ecotone. All right, don't confuse this with ecotype. All right? The ecotone is the boundary layer between our various habitats. This boundary layer can be very abrupt or it could be very gradual. All right? And that has a role in the intraspecific variation. So, I'm going to have to use a small one. Assume we have 2 patches. Patch A, patch B. This is inhospitable. All right, so our populations are in that area. Let me increase this. All right, our species can live in either patch. Let's say that these patches are very different. All right, different letters, very different. We're going to see a lot of intraspecific variation. They can move between the patches. We'll say that they can move. They just can't live in this space. All right. This represents a very abrupt change, which could lead to very drastic differences. But let's say our patches are closer. Let's see this. And now we have this zone where our species can live. It's no longer inhospitable. All right? Our zone represents this ecotone, that transition area. All right? In this case, we have an abrupt change because that ecotone was very, very small. All right? This ecotone is definitely not an area where probably our species prefers, but it can live. All right? We've got A and B here, very different patches. But our organism here can easily live in here, but as it travels, it sees a very abrupt change. So in this ecotone, with this abrupt change, definitely we're going to see a lot of intraspecific variation. But if we have something like this, what's going to often happen is we get a gradient between a gradient between our patches. We have a very large ecotone. Our organism isn't going to see this abrupt change. As it moves, as it travels, it sees more of a gradual change, and that gradual change starts to minimize the intraspecific variation that we see. So big differences, a lot of variation. Small differences, not much variation. If we have a large ecotone that helps to modulate those differences, we have small differences. If we have a narrow ecotone and thus have a very drastic change in our selection pressures, we'll get an increased intraspecific variation. Questions? So magnitude of differences, that ecotone kind of establishes our gradient. Isolation. Isolation plays a role. So we've kind of said that our species can kind of move. But if we look at this and say, if we're close, we can move, but if we're not, if we're far away, we can't, that can also play a role in our intraspecific variation. We kind of talked about this, right? We talked about this a little bit. In terms of the forces of selection, we said selection is going to be stronger in more isolated populations. Is it necessarily stronger? No, it's not. I mean, whatever the selective force is, it could be the same, but we see the end product of selection more in those isolated populations. Why? It's because we don't have the gene flow that's happening. Gene flow helps to kind of mix in all of our variation. Gene flow helps to eliminate a lot of this intraspecific variation. So if we are more isolated, if we have two patches that are far away from each other, we can produce higher amounts of intraspecific variation. We don't have gene flow modulating or eliminating that variation. Close patches, you're going to readily have gene flow. So as evolution tries to take you down one path, gene flow is countering that. So you can kind of go back and look at our evolutionary ecology presentations to see those. And the relative size of the patches. How big are the patches? And for this, we're talking about how big are these patches relative to the movement of our organism. Right? This is basically a concept called grain. So we can have a coarse grain variation or a fine grain variation. And you can think of this as kind of resolution, TV resolutions. These names went back to the day of film, like actual film that you put in a camera. All right. So you had different speeds of film. Speeds were related to the grain size on that film, and sometimes you see grainy pictures, sometimes you see crystal clear pictures. Today, I kind of talk about, high definition video, so ultra high definition videos versus standard definition TV, which you don't really see standard definition unless you go to, let's see here, you guys probably go to your grandparents' house and they probably pull out, oh yeah, I've got the movie, and they pull out a VHS tape if you've ever seen one of those. Yes, yes, I assumed you had, Kyle. Low definition. All right, so what's coarse grain variation? In coarse grain variation, our spatial variation is very large relative to the movement of our organism. All right, so in our diagram here, each of these cross hatches represents a patch, and the lines represent a different species. So a species that sees its environment as coarse-grained means that it typically only sees one type of patch throughout its life. So a snail in a forest sees the forest as a forest. It doesn't see the edge habitats. It doesn't see the field that's right next to the forest. The only thing that it sees is a forest. So for a snail, its movement pattern is pretty small relative to that spatial variation, and thus we refer to that as coarse grain variation. Now, for this type of variation, selection tends to produce the best fit phenotype because our organism only ever encounters one type of habitat patch, or possibly two, all right, few numbers. All right, so selection can produce a best fit phenotype. Fine grain variation, on the other hand, is when our patches are small relative to the movement of our organism. Consider a hawk in this same area. That hawk can fly. It can stop. It can feed in all these different habitat patches. So our hawk experiences more than one patch throughout its life, and in many cases, just across a normal day of foraging. All right? So our organism's going to see its environment as being very fine, high definition. It can see all the detail that's there. In terms of evolution, evolution can't really select the best fit phenotype for any one habitat because our organism doesn't spend a whole lot of time in an individual patch. It can move and go experience another patch. So what evolution tries to do is to look at all these patches and says, what's going to be the phenotype that allows us to survive and at least do the best across all patches? You might not be the best in any one patch, but at least we're going to be good enough to survive and outcompete others that are in the same situation. All right, so evolution tries to balance our fitness across all of our patches. So which one leads to more intraspecific variation? Those organisms that see its environment as coarse grain variation, or those that see its environment as fine grain? What do you think? Which one's going to have more intraspecific variation? Of those here, who votes for coarse grain variation? Who votes for fine scale variation? All right. Who is undecided and votes third party? All right. So in this case, we have to think about intraspecific variation. All right. You have to think about the species. We can have populations of snails in each habitat patch. All right. And each population then is going to experience its own selective force that pushes it down its own evolutionary trajectory. So we have the possibility then of getting increased phenotype, increased genetic variation when dealing with this coarse grain variation. With the fine grain variation, we're balancing across all habitat types, all patches. So what we tend to do is kind of normalize or neutralize a lot of that variation. So yeah, coarse grain variation, that should produce more intraspecific variation across the species because we could have population A here, population B, population C, population D, population E, and each population has its own phenotype, has its own genotypes going forward. Whereas our hawk sees this fine grain environment, that's really just population A throughout this entire area. All right? All right, so what we'll do is we'll pick up temporal variation on Friday. We finished out the aquatic environment, so environmental variation in aquatic systems. That quiz will get released. You'll have by the end of the day on Friday to get that quiz done. Stay up on it. All right? Once it gets released, you should get an email from Blackboard. All right.